also increases the number of calcium ATPases I mentioned just now, and that results in calcium absorption. That is the dietary calcium we are talking about. And it facilitates the calcium reabsorption in the kidney, I said, by increasing the TRPV fuels and increases the activity of the osteoblast, therefore necessary for calcification of the matrix. I hope this has gone into your mind, the mechanism of acting on the kidneys as well as it is acting on the bones. And when I said bones, I said stimulation of osteoblast. It brings secondary increased activity in the osteoclast. When we say all this, then which there is no response to vitamin D. Gone now. Okay now. So how it is? The inactivation mutation. I said that the vitamin D is synthesized. I think it's gone back. Wait a minute. Just wait a minute. Okay, we were here on calcium, and we said that the inactivating mutation of the 1-alpha hydroxylase. If you recollect, I had mentioned that vitamin D3 is synthesized in the skin by 7-dehydrocholesterol under the effect of ultraviolet rays, and this cholecalciferol is entering into the blood and going into the liver, enzymes in the liver, which converts it into 25-hydroxylase cholecalciferol. And this 25-hydroxylase, a enzyme which is known as 1-alpha hydroxylase. I have discussed it in the last lecture of mine, which converts into dihydroxy 1,25-dihydroxy cholecalciferol, and that is the increased, or that is the active vitamin D3. So, as and when there is a mutational inactivity of 1-alpha hydroxylase gene, the gene Can you hear me or not? Hello? Hello? Are you there?
Yes, sir.
Hello? Can you hear me? Can you see the
Yes, sir.
slide? Suraj?
Yes, sir.
Okay, okay. So 7-dehydrocholesterol, skin and the ultraviolet light, and then there's a vitamin D3 from the skin or also from the diet, and this vitamin D3. One is from the skin. So very important. Every day, 15 minutes sunshine or sunlight. Okay. Okay. So, from the ultraviolet light, that is the ultraviolet sun rays, that is the UV light in the skin, 7-dehydrocholesterol. And here we must keep in mind that the darker is the skin, more is the melanin. So therefore, the formation of vitamin D will be lesser and there is diet, it enters into the blood and vitamin D3 in the liver, and in the liver it is 25 hydroxy D3 and then of course 125 dihydroxy D3. I hope this is now clear, Anna. Vitamin D, we have just now discussed about it. We have discussed the target genes of the vitamin D. I said that the calcium binding protein, the calcium pump, the osteocalcin, and 24-hydroxylase, all of these genes, they are increased. Whereas it regulates the 1-alpha hydroxylase, it also regulates the parathyroid hormone, and therefore their transcription is decreased. Okay. Vitamin D action we had discussed, we have discussed all this, we had discussed all this osteoblast and proper bone formation. We did say it's Sana, which I mentioned somewhere here, that is the growth abnormal in the rickets. There is a bowing of the legs and the bone deformity in the rib cage that also is known as the also there is a abnormal growth in the wrists as well as in the ankles. And we did mention about the osteomalacia, which is in the adults, and there is a poor mineralization of the newly laid osteoid. And there is a pseudo fractures which are visible on the X-rays. Weak bones and therefore bone pain and bone fractures are very common in cases of osteomalacia. So I think that's about the vitamin D. I hope this is clear, and if there is any queries, then you are most welcome to ask me right now, and we will proceed on to the parathyroid hormone. Parathyroid hormone is synthesized and secreted by the parathyroid gland. I did mention about it. They are lying posterior to the thyroid gland. And the cells, they are the principal cells which are secreting. They are known as the chief cells. They are the principal site of PTH synthesis. It is the measure of calcium homeostasis. When the word used major means it is the most important hormone for calcium homeostasis in the humans. There are other cells also, but they are known as oxyphil cells, but their function is not known. What you are to remember is parathyroid gland synthesis and major hormone for calcium homeostasis. Okay. Here it is 4 parathyroid gland behind the thyroid gland. And synthesis. PTH is translated as a pre-pro-hormone cleavage of pro sequence, or you can say pre-pro hormone molecule, and then it is cleaved, and the biologically active peptide PTH is having the dominant regulator of PTH is the plasma calcium. Secretion of PTH is inversely related. When I say inversely related, means low calcium levels, more PTH production. More calcium levels, less PTH or the parathormone production. So remember this much only, that the dominant regulator of PTH is the calcium, plasma calcium, and secretion of PTH is inversely proportional. And this is, of course, perhaps if at all these plasma levels of calcium, they are going down, let's say in a hypocalcemia, we did say that the levels of calcium normal are 9 to 11 milligram percent. And if there is a less than 8.7 milligram percent or so, that is hypocalcemia, and hypercalcemia calcium level is more than 10.4 milligram or so. And these are the levels which can increase the physiological changes. And what are these physiological changes? We did discuss that they are neuromuscular dysfunction, there could be CNS dysfunction, there could be renal insufficiency and soft tissue calcification, as well as skeletal pathology. So I said there are 2 types of cells. This is the regulation of PTH. I will just come to it, but let me say that the chief cells, they have prominent Golgi apparatus as well as endoplasmic reticulum. So that means that something is happening in the endoplasmic reticulum as well as in the Golgi apparatus, and then this so therefore it is synthesized and secreted. Large and abundant oxyphil cells, I said, their function is not known, they are also present, but they are rich in mitochondria. So we are talking about the principal cells or the chief cells which are secreting PTH. They have got a prominent Golgi apparatus and the endoplasmic reticulum. Human PTH, the molecular weight is 9,500. I said it has got 84 amino acids. I said it is synthesized as pre-pro-parathermone. And this pre-pro-parathermone has got almost 100. They are rich in endoplasmic reticulum. So therefore, in the of amino acids is removed from this pre-pro-PTH to form pro-parathermone, which has got around 90 amino acids. And then in the Golgi apparatus, Another 6 amino acid, they are removed, and 84 amino acid PTH, proparathyroid, is packaged in the secretory granules and is released as a product of the chief cells. The half-life of parathyroid is only 10 minutes, and the secreted polypeptide into the fragments. When I say half-life, naturally, this PTH must be degraded, and this degradation occurs by the Kupffer cells or in the Kupffer cells. So these fragments, they can be cleared by the kidneys. How do we measure the PTH levels? And I think biochemistry must have discussed it, but we must say that it is the immunoassay technique which measure the mature PTH 84 amino acid parathyroid with it the mature, that only can be measured. Okay. So regulation of PTH, we said secretion respond to smoke. There is a unique calcium receptor in the parathyroid. This senses the changes in the extracellular fluid. And what type of receptors are these? These are G protein. Activate phospholipase C and inhibits the adenylate cyclic AMP. Okay, we did mention about the second messenger. If you recollect, I had mentioned about the second messenger, and I said these second messenger to the specific sites in the cytoplasm or the nucleus. And they are specialized messengers which amplify the signal. And we did discuss there are three molecules. One is the adenyl cyclase or procyclic AMP system. And second is the calcium comodulin, second messenger I did mention. And third, of course, is the cell membrane phospholipid, second messenger. And here we are talking about the same typical G protein coupled receptor that activate the phospholipase C as well as inhibit the adenyl cyclase, and that results in increase in the intracellular calcium by generation of inositol phosphatase, and that decreases in the cyclic AMP, which prevents exocytosis of the PTH from the secretory granules. This is being made clear further up. When calcium falls, cyclic AMP rises and PTH is secreted. Cholecalciferol or vitamin D3, therefore providing another level of feedback control for the PTH. We did mention at that time that vitamin D3 is being activated by, there is no direct control of PTH, is excreted by the phosphate level. Anyway, you have to simply remember this, that calcium falls, cyclic AMP rises, and PTH is secreted. Okay. This is what I was discussing, that there is a cyclic AMP, adenylase cyclic AMP, and phospholipase C. Let us talk, we talked about the calcium low levels. When there is a calcium, they are activated. And what it does, it activates the adenyl cyclase, plus activates the adenyl cyclase, but it inhibits the G protein receptor, that is the phospholipase of the PTH. If the same thing we apply cyclase CMP system is inhibited, it is inhibited, and phospholipase C or the is stimulated. So therefore, there is no secretion of calcium here and therefore no secretion of parathyroid. We had known that calcium is a messenger for secretion of transmitters as sensitive receptors. They are working by two ways. One is the adenyl cyclase CMP, cyclic AMP system, as well as by phospholipase C, and it responds to the calcium levels. If the calcium levels are high, it is the phospholipase C system is stimulated and adenyl cyclase CMP, cyclic AMP is inhibited, and therefore PTH levels are lowered. And if there is a calcium low level, it is opposite, that the adenyl cyclase cyclic AMP system is stimulated and therefore there is a increased production of parathyroid, whereas this phospholipase C is inhibited, releasing more calcium and therefore increase in the parathyroid. I hope this has made a bit clear by me and how these calcium sensitive receptors are working on the calcium sensitive receptor. They are sensing the calcium levels in the extracellular fluid. Shreya, Shrestha Shreya, does that make a bit sense? Hello.
Yes, sir.
Shrestha Shreya, huh? Through your books and try to understand a bit at length. So PTH action, overall action of the parathyroid is to increase the plasma calcium levels and decrease the plasma phosphate levels. And we had discussed in detail earlier how PTH is acting and how does it increase the plasma level. Number one, it acts directly on the bones to stimulate the calcium resorption. It acts on the kidneys to stimulate the calcium and it inhibits the reabsorption of phosphate. If calcium reabsorption, the kidney is stimulated and the phosphate is excreted. When I say inhibit reabsorption, that means phosphate is increasingly secreted. And we had discussed that PTH also indirectly acts on the intestine. Now here the you need to understand, here it is, that direct action is on the bones for calcium resorption and direct action is on the kidneys for calcium. Indirect action of PTH on the intestine hydroxylase enzyme in the kidneys, therefore increasing the synthesis of 125 dihydroxycholecalciferol vitamin D3, and we had discussed in detail that vitamin D3 is the one which is responsible for increased Calcium versus PTH. I think urine calcium excretion and plasma calcium levels. If the plasma calcium levels are low, naturally the urine calcium excretion is less. That means absorption is much more. And if PTH levels, plasma calcium levels, they are much more, then their excretion is much more. Action of PTH on the bones, though we have discussed in detail in my earlier lectures, but PTH acts to increase the degradation of the bone, release of calcium. It causes osteoblasts to release the cytokines, which stimulate the osteoclast activity. We have talked in detail about the cytokines, something like interleukin 1, interleukin 6, tumor necrosis factor, TNF factor, and so on and so forth. It stimulates the bone stem cells to develop into osteoclast. We have discussed factor. If you recollect, I had discussed in detail. So that stimulates the bone cells and develop into osteoclast. And the net result is increased release of calcium from the bones. This is an important topic you must understand. Go back into my earlier lecture, go back to the books and read that how does PTH acts for degradation of the bone or for resorption of the bones and release of calcium. Effect on bone are dependent upon the presence of vitamin D, and we have discussed this in detail. Action of PTH on the kidneys, acts on the kidney to increase the resorption of calcium, we just now discussed. And it gets increased excretion of phosphate. We said inhibits phosphate reabsorption. That means it increases the excretion of phosphate. And get increased production of the active metabolite of vitamin D3 because it stimulates 1-alpha hydroxylase enzyme in the kidneys, which is responsible for converting 25-hydroxycholecalciferol into active 125-dihydroxycholecalciferol. And the net result is, of course, plasma calcium level. And I think this is a beautiful diagram which shows you. Ishika, you are there?
Yes, sir.
Understanding?
Yes, sir.
Okay. So here it is a hypocalcemia, that is the low blood calcium which stimulates the parathyroid gland. This side is the parathyroid gland releases the parathormone, PTH release from the parathyroid gland. It acts upon the bones first, right? What does it do? PTH activates the osteoclast. Calcium and phosphate ions are released into the blood. This side is all blood. This side is all blood. This side is the PTH goes and acts upon the intestine. But if you look down here, PTH goes to the kidneys, and in the kidneys it increases calcium reabsorption is 1, and it also promotes activation of vitamin D. And this activated vitamin D3 or 125 dihydroxycholecalciferol is the one which is responsible for increased calcium absorption from the food, and here is the entry into the blood. And as the calcium levels are rising, the PTH release is inhibited. The feedback control. I think this is a beautiful diagram, Anna, which tells everything about hypocalcemia, its action on the parathyroid gland, the release of PTH on the bone, on the intestine, on the kidneys. This is direct action, this is a direct action, this is indirect action by synthesis of vitamin D3. I hope this is made clear, and your mechanism of action of PTH binds to G protein coupled receptors. Binding of PTH to its receptors activate two signaling pathways. I discussed it earlier, Anand. Increases the cyclic AMP, increases the phospholipase C, activation of phosphokinase A appears to be sufficient to decrease the bone mineralization, and both A and C, they activate this much on G protein coupled receptors, and two pathways. One is cyclic AMP and second is phospholipase C, and that was beautifully explained in my earlier diagram. Regulation of PTH we have been discussing, but response to change in the calcium levels, low calcium level result in high PTH, high calcium results in low PTH. And PTH cells contains a receptor for calcium coupled to a G protein. We did mention that chief cells, PTH cells, chief cells, they are having a calcium sensitive receptors, and that is a G protein coupled receptor. A result of calcium binding, we said if there is this is what is being explained. And vitamin D, of course, inhibits PTH release. Calcium receptor, cyclic AMP and PTH release, this perhaps Anna is telling you, and if calcium is there, the AMP, and then of course decreased PTH. More calcium, decrease in cyclic AMP, and decrease in the, and here it is a no calcium, increased cyclic AMP, and there is increase in the release of PTH. I hope this makes it very clear. PTH related peptide, there are certain peptides. I will just continue for another 5 minutes. Let me finish it here, because of the interruption which has been going on, has a high degree of homology to pH. They are very much, these proteins, they are very much similar, but is not from the same. It can activate the PTH receptors, and in certain cancer patients with high PTH related peptide levels, this peptide causes hypercalcification, kidney, glomerular function, and of course growing and development. I think I will leave it here, and we will go in my next lecture, that is tomorrow, about the primary hyperthyroidism. Please do not leave. Let me take your roll call first. Okay, then here it is, and